seen in the differences between in human DNA sequences, the evolution of genes, and the variation of the genomes. How genes are, uh, how variation happens in the genomes. If you take the examples of here, the sequence of the nucleotide sequence of the one DNA or one gene, then if SNP, SNP means single nucleotide polymorphism. If mutation occurs in a single nucleotide, if mutation occur in in adenine, then uh, uh, three nucleotides codes for one amino acid. Suppose ATA, it codes for only one amino acid. If mutation occurs at this A, then this CAA code for another amino acid. If this mutation occurs in this adenine nucleotide, then this frame will be shift. Then this mutation causes the variation of the genomes, and in evolution, this variation causes evolve of new species. Then
Sir,
if we
PPT is not visual.
PPT is not visible.
Yes, sir.
No, sir.
Okay, okay, okay. Sorry. Visible?
Yes, sir.
Yes, sir.
Yeah. Huh. We discuss about the diversity of the genome. How diversity happen in human genomes. If we go to the diversity of the genomes, this diversity of genomes can be seen in the differences between human DNA sequence. How this human DNA sequences varies by evolution of the genes and the variation of the genomes. We take the a small example of single nucleotide polymorphism. If we go to the this gene sequences, then here if we mutated one adenine nucleotide, then this frame means what? Now three nucleotide, it codes for one protein, one amino acids. If you mutated one adenine nucleotide, then this frame will shift, and this frame shift mutation causes a different kind of proteins, and this different kind of protein leads to give rise to a variation in the species. Then if you go to the insertion, if we add one nucleotide, then the same thing will happen. The gene sequence will be changed. If you go to the deletion, if you delete one sequence of the nucleotide, the same frame will shift. Means what? Now if you go for an example, if you go to the these two sequences, suppose here is ATA, it codes for a uh, amino acids. If you mutated this A, then it converted to ATC. Then this ATC gives rise different amino acids. These different amino acids give rise to a different proteins, and this gives a new species. How this diversity of genomes happens during this evolutionary process. Then this is called small scale because of the mutation in a single nucleotide, either insertion of one nucleotide or deletion of one nucleotide, or polymorphism of one nucleotide. If you go to the large scale changes during this evolution process, we know the different type of recombination occurs in our genes during meiosis process. But during meiosis, if you delete one, if you if delete one part of this genome sequences, then this give rise to a different proteins, and these different proteins then in evolutionary, it changes and evolve a new species. If you delete one gene segment, or insert a new gene segment or duplicate a new gene segment. If you duplicate, what happened? If you duplicate, unnecessary protein will synthesize. Those proteins are not required for our body. Excess protein expression. This also leads to the changes in our uh, species. If you invert the sequences of the this genome, this will be the same cases. And translocation, during Robertsonian translocation, the deletion of the uh, this recombination of this gene also give a diversity of the genome. This diversity of the genome give rise to uh, a in evolutionary process, it gives rise to a new species. Then, if we go to the C value paradox, what is C value paradox? C value paradox refers to the unexpected lack of correlation between the genome size of an organism. For an example, if we compare with E. coli genome with human genomes, how this complexity happen, and its perceived biological complexity, specifically organism with similar level of complexity often have wildly varying amounts of DNA, and some simpler organisms can have much larger genome, more complex one. It says, for an example, if we go to the genome size, this genome size doesn't equal the complexity. If someone have large genome size or a small genome size, this doesn't equal the complexity of the organism. If we go to the larger genome size, do not always mean more complex organisms. For instance, some amphibian and plants have much larger genomes than human, but they are not more complex than humans. Then if we go to the non-coding DNA, as we previous class we describe about this non-coding DNA, that is intron parts of genes. Then this much variation in genome size is due to the non-coding DNA, which is, for an example, as we discuss about introns, repetitive sequence, transposons, these are non-coding DNA part. These part of the genome doesn't give rise to any proteins. So these introns and transposons uh, they are do not directly code for proteins. If you go to the gene count mismatch, what is gene count mismatch? Many organisms with large genomes do not have significantly more genes than those with smaller genomes, but their genomes may contain vast amount of non-functional or regulatory DNA. This give rise to the C value paradox or complexity of the DNA. So the C value paradox highlights the fact that a large portion of the genome may not be involved in coding or functional traits, because if you go to our genomes, in our 100% genome, only 2 to 4% give rise to protein, other are non-functional. So this genome may not be involved in coding or functional traits. So questioning the direct relationship between DNA amount and organismal complexity. So these are the main reason of this complexity of our human genome, because only 2 to 4% are coding sequences. All the 96% are or introns or transposons or repetitive DNA sequences. Then if we go to the what is then if you go to the what is transposon, the transposons also known as jumping genes. How these jumping genes are on eukaryotic system, or these are called transposable elements. These are DNA sequences that can move from one location in a genome to another location of the genome. These transposons in eukaryotics are of two types. One is retrotransposon and second one is DNA transposon. If you go to the class 1 retrotransposon, this is a donor DNA. This donor DNA then transcribed to converted to in an mature RNA intermediated, and this RNA intermediate then reverse transcribed with reverse transcriptase enzyme to form a DNA intermediate. This DNA intermediated then integrated into the target DNA. This is the due to this uh, RNA to converted to into DNA, the reverse transcriptase enzyme is used. That's why this is called retrotransposon. Then if you go to the DNA transposon, then the donor DNA, then excision of this donor DNA transposable element, this DNA intermediated, then integrated into the target DNA. This integration of target DNA, this integration happen in the target DNA. This transposable element is directly transferred to the donor to the accepted DNA. But here, the this retrotransposon transposable element is converted to RNA intermediated. Then, in the help of reverse transcriptase enzyme, it converted to DNA intermediated, converted RNA to DNA. Then this DNA is integrated into the target DNA. Then, if we go to the other elements of this transposon, these are short interpressed nuclear element, that is signs, are class of DNA elements that are non-coding, repetitive, and transposable. These are non-coding. Non-coding means these sign region of this DNA are not codes for any proteins. These are repetitive, means in its DNA sequence, this genes is repeatedly found in an interval, and these are transposable, means that these are jumping genes. These transpose from one donor DNA to the targeted DNA. Then they are a type of retrotransposon. These sign molecules are called these class 1 retrotransposon molecules, which are DNA elements that can copy and paste. These are called copy and paste themselves into the regions of the genome. Then line. This line long interspersed nuclear elements, also known as nucleotide elements or long interspersed element line, are a group of non-LTR retrotransposon. Same. These two are sign and line are retrotransposon class molecules or genes, are widespread in the genome of many eukaryotes. These are found, line and sign elements are found throughout our non-coding. 96% of the non-coding region, these are mostly found, line and sign. Then these lines are the most abundant transposable element within the human genome, with approximately 20.7% of the sequence. These identified as being derived from lines. The only active lineage of line found with human belongs to the line 1 class and is referred to as LNH. These are widespread in our genome, these line elements, and the human genome contain an estimated of 1 lakh truncated and 4,000 full length line 1 elements. These are truncated, means these are not functional, and 4,000 full length line 1 element due to the accumulation of random mutation. Random mutation means any mutation happens in our coding or non-coding part of the gene. Then this sequence, many lines has degenerated to the extent that they are no longer transcribed or translated. A comparison of line DNA sequence can be used to date transposon insertion in the genome. These line DNA sequences are the transposon insertion in the genome because these are jumping genes. Then Next transposition is ALU elements. And these are highly repetitive DNA sequence. Due to this highly repetitive DNA sequences, these forms a loop-like structure because due to the repeatedness of the same sequence, they form internal hydrogen bond. So this loop structure will be formed, and that can be classified as signs. And these are also a part of also called as sign, which are themselves a type of non-autonomous retrotransposition. These are non-autonomous retrotransposition, and these ALU element have a left monomer and a right monomer, represent two halves of ALU sequence which similar to each other. These sequences are similar to each other, and the A-rich tail, this A-rich tail consists of several adenine bases at the end, and the function of this ALU element is gene regulation. ALU element may affect gene expression by inserting into promoter regions or exons. If it inserted in the promoter region or exon part, because exon are our protein coding region, if this sequence will be inserted in that part exon region, the gene will be not functional, so gene regulation will be happen. If you go to the evolutionary role, they contribute to a genetic diversity and genome evolution through their copy paste mechanism. Due to the changes in it gives rise to a different changes or in our gene. So it give rise to a new species by inserting or deleting any nucleotide bases. And these insertion and deletion may causes different types of genetic disorders, including cancer and neurodegenerative disease, when they disrupt or in functional genes. Then here anyone have doubt these transposable elements?
ଯୋଉ ALU ଏଲିମେଣ୍ଟ ଯୋଉ ଗୁଡାକ କହିଲେ ସେଇ ALU ଏଲିମେଣ୍ଟ କୋଉ ଗୁଡାକ?
Signs ALU, this is the structure of ALU element. These are non-autonomous DNA.
Yes sir.
Okay. Then we'll go
Sir,
for
sign ର full form କଣ? SAINAE sign.
here we have discussed short interspersed nuclear element, that is sign. These are short
Yes, sir.
sequence. They like this. These sequence are inserted into the from a donor to the targeted DNA.
Sir, ଆଉଥରେ short intersperse.
Interspersed nuclear
Intersperse
element.
nuclear element. Okay, sir.
Then we go to the gene duplication. Duplication is a mutation that produce duplicate copies. One copy is there, then another copy is formed. These duplicate copies of genes, which serve as a raw material for change during the evolutionary process. If one suppose X gene is there, and X gene is give rise to a Y protein, which which is the building block of our body. Suppose excessive Y protein, then this gene is duplicated, and this X gene give rise to one Y protein, then another duplication of this X gene is give rise to Y protein. This Y protein, excessive Y protein, doesn't required for our body. For this, the the evolutionary changes give rise to the complexity or evolution of this new species. Then if you go to the when a gene is duplicated, the extra copies are functionally unnecessary to the organism, leading to functional redundancy. In some cases, and this expression, hyper expression or excessive expression may lead to causes different types of disease like neurodegenerative diseases and cancer. These causes also cancer. And in some cases, the expression of the additional genes may even result in an imbalance of gene products. In many cases, the duplicate genes are therefore made non-functional by deleterious mutation or splicing error, give rise to non-functional gene or pseudogenes. If these duplicated genes uh, this duplicated gene is non-functional, so this give rise to a non-functional protein or error protein, and this error protein or non-functional protein may be give rise to different types of diseases like neurodegenerative diseases. For an examples, Alzheimer's or Parkinson diseases, different types of deposition of different alpha helix or beta acids in our brains. So these gene duplications are of four types. One is tandem duplication. In tandem duplication, the tandem duplication usually takes place by the homologous recombination between repeated sequence such as ALU sequence involve unequal sister chromatid exchange. During this meiosis process, if tandem duplication occurs between the mm, tandem duplication occurs between the genes and the ALU elements, and it give rise to gene copies which insert in tandem at a chromosomal location. For an example, tandem gene duplication gave rise to a paralogous alpha and beta globin gene, which lead to evolution of multimeric hemoglobin composed of two subunits of 2 alpha and 2 beta subunits. This tandem duplication will occur between the gene and the ALU elements, and this give rise to the evolution of the different alpha and beta subunits of the hemoglobin. Then if we go to the second one, that is transposon mediated duplication or DNA transpose duplication. Then it is a result of DNA based replicative transposition in which gene remain in its original location while the duplicated copy is inserted at a new chromosomal location. As previously we described, if insertion or deletion occurs in a gene or inserted a new gene by this transposable element, it also give rise to the duplication of this gene. Then if you go to the segmental duplication, the segmental duplication or duplicons are regions or more than 1 kilo base pair length with their with greater than 90% similarity. One particular segment having duplicated, and these duplicated parts also give rise to the same region of that part, and these are 90% similarity and are present more than once in a genome, either on the same chromosome, that is intrachromosomal, or different chromosome, that is interchromosomal. Approximately 5% of the genome comprises of segmental duplication such as lines. Segmental duplication means large amount of transposable element is inserted in that genome, and this segmental insertion is similar to the previous genome, so that is 90% similarity. Then if you go to the fourth one, that is retrotransposed duplication, and it is the duplication of a gene via reverse transcription of processed mRNA into a complementary DNA copy which inserted back into the genome. As we here we described about the retrotransposon, how this retrotransposon occur. Then the donor DNA having the this line sequences These sequences are transcribed into a RNA intermediated, and this RNA intermediated by enzyme reverse transcriptase, it converted to a DNA, and this DNA is integrated to the target DNA. This is called retrotransposon. These retrotransposon duplications uh, give rise to a gene, that is PTEN gene is retrogene arising duplication. PTEN gene is for tumor suppressor gene. If this gene is excessively expressed and uh, this gene doesn't require for our body, then it gives rise to form a tumor or cancer in our cell. Any deletion or insertion or excessive expression of the protein may give rise to a cancer in human beings. Then, if we these are called gene duplication. Then how we'll study this type of gene duplication, genome mutation, then tandem duplication, segmental duplication? How we'll study that part? Then if you go to the transcriptomics process, transcriptomics means copy of uh, from DNA to RNA. Here we study the messenger RNA or mRNA as well as different types of non-coding RNA. Then what is transcriptomics? Because why we choose to study the transcriptomics process? Because genome is transcribed to mRNA. These mRNA will functional give rise to a functional protein, and that protein are helpful our building block or helpful or enzymes or for metabolic process for our body. So any error in the mRNA level, it give rise to a non-functional protein. So to study this transcriptomics is the study of the transcriptomes, the that is the complete set of the RNA transcript. Here we study the complete set of the RNA, means a cell having different types of transcript like messenger RNA, non-coding RNA. Non-coding RNA means micro RNA, a small nuclear RNA, hnRNA, heteronuclear RNA. These are called non-coding RNA. This what is the meaning of non-coding RNA? These RNA doesn't give rise to any proteins, and these messenger RNA give rise to a protein. These transcripts produced by the genome under a specific condition in a cell, tissue, or organism. It provides insight into gene expression patterns, helping the researcher or scientist to understand how genes are regulated and how they respond to various stimuli. Means what are the various stimuli? Means any stress condition or any disease condition, how these mRNA levels are vary, and how they are contribute to a cellular process, and how they give rise to expression of a protein in cellular process. Then, if you go to the key concept of this transcriptome, then transcriptome are the total collection of RNA transcript. These are collection of RNA transcript in a cell or tissue at a given
ମୁଁ
time,
ଏକ୍ଚୁଆଲି
because
ଦେଖେନି ନା ସେଇଥିପାଇଁ
some transcript are required.
ମୁଁ ଏତେ ଚେକିଂ ହେଇପାରିବି ମତେ ଇଷ୍ଟା ଦେଖୁଥାନ୍ତି ଏ ଦେଖୁଥାନ୍ତି ତାପରେ
Please
ଦେଖୁଥାନ୍ତି ସେ
silent your phone. Ketaki
କଣ ଦେଖି ମୁଁ
Pradhan,
ତ କିଛି
please
ଦେଖୁନି।
silent your phone. Then, what is the key concept behind this transcriptome? If the total collection of RNA transcripts, suppose for an example, we are taking an example of a one cell having cancer or we take one small biopsy of that cell, then we uh, extracted all the mRNA from that biopsy, and we study how these mRNAs are overexpressed or underexpressed in that particular biopsy. So after extraction, then we purified the RNA. Then we after purification, then we create a cDNA library by this reverse transcriptase enzyme. Then we sequence all the mRNA molecules by cDNA library, because we converted to cDNA library because RNA is mRNA is very unstable, because any change in pH or any alkali condition induced, this mRNA will be degraded. So we converted it to cDNA, and this cDNA in sequencing machine, that is Illumina sequencing machine, give rise to the all the sequence of these mRNA reads, and this give rise to the gene expression. How if the mRNA concentration of the X gene is increased, then this increase or decrease will create some disease. So we'll establish that condition is cancer or not. Then if we go to the gene expression, then transcriptomics measures the levels of RNA transcript to determine which genes are being expressed and to what extent. If we go to this RNA transcript, then this mRNA transcript having multiple genes, having multiple or millions of the genes. How will found that this is the X gene? Because after sequencing, if you blast means one reservoir gene bank is there. Here, if we target this sequence to this gene bank, then this gene bank give rise to the similarity to this X gene. How much similarity? That is 100% or 90% or 80%. According to this, we predict that this mRNA is for this X genes. Then we will study then the gene expression. These transcriptomics measures the levels of RNA transcript to determine which genes are being expressed and to what extent. If X gene have 10 transcript and Y gene have 8 transcript, this means X gene is overexpressed as compared to the control or a normal patient. How we'll study this transcriptomics? If we go to the transcriptomics, different transcriptomics techniques, then we'll study RNA sequencing or RNA-seq using this Illumina machine, that is Illumina Mini-seq platform. This is the most common method. This Illumina machine is the most common method for a transcriptomic analysis, which uses next generation sequencing to quantify RNA levels. This machine directly read all the mRNA molecules or cDNA molecules. Then next one technique is microarray. This is a technique that use probes to measure gene expression levels for thousands of genes simultaneously. Because if we label the fluorescent cDNA of sample A and sample B, then we go for cDNA microarray, because previously here we have added the X genes or Y genes sequences. Then this gene or mRNA will bind to this uh, similar sequences, because if this gene have X sequences and here we also added some genes, like we go for the um, uh, in cancerous patient sample, we go for the analysis of the grading of the cancers, because we know the target sequences, and then we added this gene sample A or sample B to this targeted sequence, if bind will occurs, and it will give rise to a hybridization fluorescence, and this fluorescence imaging data analysis give rise to X or Y genes. And this give rise, these two techniques of the transcriptomics, that is RNA sequencing and microarray, give rise to the, if the mRNA uh, is overexpressed or underexpressed, and according to that we can predict the patient will be disease or not. Then the application, what is the application? The gene function and regulation. By analyzing which genes are being transcribed in response to specific condition, researchers can deduce the roles of that genes. If we found any particular genes is responsible for that. For an example, we know if mutation occurs in the P53 protein, then it will leads to cancer, because P53 is a tumor suppressor gene. Then if the mutation have, if we go for the sample A or sample B, this sample A and sample B having mRNA of this P53 gene, then this P53 gene will be hybridized to the known sequence of the P53, and this hybridization will give rise to a fluorescence image, and fluorescence will be quantified by this imaging and data analysis machine, and this will give rise to overexpressed or underexpressed of this P53 gene. Then in disease research, transcriptomics help identify gene expression changes associated with disease like cancer, then revealing potential biomarkers or therapeutic targets. Then the major study transcriptomics is for biomarker study. In biomarker study, sorry, in biomarker study, as you described, P53 is a gene. If P53 is overexpressed or underexpressed, it will give rise to cancer. Then this will be studied by this transcriptomic analysis. Then in developmental biology, it provides insight into how gene expression changes during different stages of development. Because if you go to the we know how the gene will expressed, because we during the developmental process we can transcript transcript transcriptomics of the different tissue or cell uh, to know which gene is expressed at that particular time. So we can also analyze the mRNA of the um, genes. Then drug response. In drug response, suppose X drug is treated for the down regulation of the a particular genes which causing cancer. Then we'll analyze that particular gene mRNA, and we'll know how the mRNA is upregulated or downregulated. And this study will give rise to the uh, patient's gene expression. And these transcriptomics can identify how gene expression changes in response to drug treatment, and helping in drug development in personalized medicine. And this will help for new drug development in personalized medicine. Then, if you go to the proteomic analysis means here we are analyzing the large scale study of proteins, particularly their structures, functions, and interaction with biological system. If you go to the protein have different structures. For an example, as we know, protein have primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure is total stable by covalent bond, and secondary structure is stabilized by hydrogen bond, and tertiary structure is stabilized by disulfide bridge, that is a covalent bond, and also different type of non-covalent bonds. And quaternary structure is assemble of tertiary structure by hydrophobic interaction or disulfide bridges. Then how we analyze that part? In proteomics, we first extracted the proteins, all the proteins of the cells or tissues. Then these proteins are digested. As we know, we can digest it by different enzymes. One enzyme is called trypsin that cleaves the protein at uh, lysine or arginine residues of that targeted protein, and it will give rise to peptides, and these peptides are separated by this machine is called mass spectrometry. Here in transcriptomics, the machine is called Illumina Minisec machine, and here the machine is called mass spectrometry. And this mass spectrometry analyzes the whole protein and give rise to the protein name. And we can also we can do by this protein, we can, if it targeted, we can do targeted proteomics by immunoprecipitation. Suppose we want to extract only immunoglobulin, then we can do the immunoprecipitation by immunoglobulin antibody. Then we separated by size exclusion chromatography or sodium dodecyl sulfate polyacrylamide gel electrophoresis. Then we can do the peptide separation, then mass spectroscopy analysis, and this mass spectroscopy analysis give rise to the different proteins. Then these proteins are the functional molecules in our cell, because all the proteins are functional molecules, or all enzymes also are made up of proteins which are metabolic active, because in our cell many enzymes or proteins are involved in signaling molecules or metabolic processes. These are very important parts of our cell, and uh, these responsible for nearly all cellular processes, and studying them helps in understanding how genes are expressed at the protein level, because ultimately all genes are expressed in the protein level, because all proteins are in signaling or enzymatic in nature. All enzymes are made up of proteins, and these enzymes are functional element of a cells, and these identification may help in different disease analysis, like cancer, neurodegenerative diseases, or different types of metabolic disorders. We can analyze by this proteomic analysis. Then, if you go to what is proteome, proteome are the entire set of proteins that expressed by a cell or tissue or organism at a certain time under specific condition, because specific condition is mandatory. If a person having in stress condition, the protein concentration will also vary. A person having different kind of uh, and they have different type of food condition or climate condition or any type of environmental condition, that also leads to changes in the proteome of that of particular organism, because different organism have different proteome levels. Then we can study the post translational modification, because this is the important process, post translational modification, because this post translational modification may be give rise to silencing of that protein or genome. because if acetylation will occur or lysyl lysine acetylation will occur or methylation will occur, any add of the any functional group to a protein may be a dysfunctional protein, and this dysfunctional protein having no role in signaling molecules. So we can study the post-translational modification of protein also. This protein undergo modification after translation, such as phosphorylation or glycosylation. Phosphorylation is very much important process because phosphorylation or dephosphorylation of a protein may regulate the activation or inactivation of any enzyme, because we know a kinase is an enzyme, it gives rise to add a phosphate group, because phosphorylation, if phosphorylation happen, then that enzyme is activated. If this the dephosphorylation will occur, then the enzyme is deactivated, or any glycosylation or any addition of a sugar moiety to a protein. So this is also important glycosylation process. Then we can study how phosphorylation is occur or not, glycosylation is occur or not, because these are required for our cell. So this which affect their function and also regulation. This also controls the function and regulation of the cell. Then how we'll study this proteomics? We can study by the proteomics by mass spectrometry analysis. What is mass spectrometry analysis? A primary tool for identifying and quantifying proteins and their modification. By this mass spectrometry techniques, we can identify the protein and also their post-translational modification. We can use two-dimensional gel electrophoresis. These techniques, this RNA sequencing and microarray, and this mass spectrometry and 2D gel electrophoresis or protein microassay, will discuss it later. We'll discuss it later. By this two-dimensional gel electrophoresis, we can discuss about this SDS page electrophoresis. Then we can discuss the two-dimensional gel electrophoresis. This is used for separating protein based on the charge and size. Here we can separate the protein according to their charge and size. If a larger protein move slower, because retardation will happen. If a smaller protein goes faster, because less retardation. So according to their charge and their size, then this protein also will separate. And in protein microarrays, we also we also here we described about the microarray of like DNA microarray. Here also we can have protein microarray used to study the protein interaction and function, because as we know ligand receptor binding, this protein will occur or not, we can study this in protein microarray. In we isolated the ligand, then in our the microarray we can give the ligand, then this ligand and receptor, sorry, in the microarray we have receptor and we isolated the ligand, then we give rise the ligand to the this receptor, and this ligand or receptor will bind or not, we can study that part also in protein microarray. And the application of this proteomics techniques is, first one is biomarker discovery. And this if you know if you know the particular which protein is causing cancer, then we can discover the biomarker discovery by proteomics. This proteomics technique helps to identify protein biomarker for diseases, particularly in cancer, different types of cardiovascular disease, and neurodegenerative disease. Different type, any type of disease, you can use this proteomics techniques for biomarker discovery. Then we can target by drug development It aids in understanding how protein interacts with the drugs and finding a new drug target. This if we target some drugs to that proteins and that protein is down regulated or up regulated, we can find this by these techniques by mass spectrometry. And in the functional study, proteomics allows for the study of protein networks and pathways. And if you know which proteins are involved in the disease processes, then we can study that protein pathways, and this pathway revealing how proteins work together in biological system. And also in we can study the post translational modification. This post translational of PTMs helps understand protein regulation and cellular signaling pathways. Like this is the omics approach, that is genomics, that is in DNA levels. Epigenomics means that is in our histones level. In transcriptomics, that is mRNA level. In proteomics, at study at protein levels. And in metabolomics, that is metabolized, that is signaling or small molecules. These techniques will start these techniques give rise to study different types of diseases, and this will occur by this genomics, epigenomics, and transcriptomics by next generation sequencing, and this proteomics by mass spectrometry analysis, and also metabolomics also this mass spectroscopy analysis. Then if we go to the genome analysis techniques, how we analyze the genome. First is DNA sequencing. We can sequence all the DNA, like all the cell DNA or tissue DNA, by next generation sequencing, that is called NGS. It allows high throughput sequencing of entire genomes or specific genomic regions, this providing a detailed map of genetic information. This machine sequences all the nucleotides. Suppose, for an example, if we go to the gene insulin, insulin gene having 51 amino acid sequences. This by this next generation sequencing, we can sequence insulin genes within fraction of seconds, and this sequencing provides any mutation occurs in this insulin gene or not. This will provide by this next generation sequencing technique. And another one is Sanger sequencing. This is a traditional method used for smaller scale sequencing tasks for validating result from NGS. If we study a small part of the gene, then we go for Sanger sequencing. If we study the multiple genome analysis or gene analysis, then we will go for this next generation sequencing. We also study by polymerase chain reaction by PCR. If we target particular gene by particular using particular primer which amplify the total DNA sequences by this PCR machine, and this give rise to the different condition. During this COVID time, we use QRT PCR, that is quantitative real time PCR. In this quantitative real time PCR, we targeted a particular gene of this SARS-CoV virus. Then this gene is upregulated in your body or downregulated in your body. We can study by this polymerase chain reaction. Everyone knows about the real time PCR. And we can study by genome wide association study. What is genome wide association study? It identifies genetic variants, because it identifies if a particular gene having changes in hierarchically or not during this evolutionary process. These are associated with specific disease by scanning genomes from many individuals. If we analyze a particular region, suppose X gene is in India, is vary from American species because they are genomic or evolutionary variation or SNPs. Then we also described the microarray processes. The DNA microarray can simultaneously examine the expression of thousands of genes or detect genomic variants by hybridizing DNA to the probes on a chip, or new technique that is CRISPR-Cas9 genome editing technology that allows for targeted modification in genome, enable functional study of specific gene, because this Cas9 enzyme guided by these CRISPR sequences to the target gene, and it will silent or chop that targeted gene, and it will doesn't give any specific product or functional protein. So by these techniques we study the functional analysis of the genes. And another one is fluorescence in situ hybridization, that is FISH. the, we can use fluorescent probe to detect the presence or absence of specific DNA sequences in chromosomes, often used in clinical diagnostic or cytogenetic study. If we target by particular fluorescence probe to a gene, and that gene is highly expressed, and it will give rise to more fluorescence than that gene in low express in a particular cells. By these techniques we can also study the expression of the genome. Then uh, in proteome analysis technique, first one is mass spectrometry. In mass spectrometry, that is standard mass spectrometry, it identifies and quantifies protein by measuring mass to charge ratio or ionized protein fragment. As we described, large protein molecule move slower, as well as small protein molecule will move faster. By this so movement, mass to charge movement, we can detect particular protein molecular weight, different molecular weight protein. And in also in matrix assisted laser disruption ionization technique, in this technique we also study the proteins and measures also their mass for identification. We can identify how what is the molecular weight of the particular protein by this technique. Then two-dimensional gel electrophoresis. In two-dimensional gel electrophoresis, we separate the proteins based on their isoelectric point. What is isoelectric point? Where the net charge of the protein is equal. That is equal number of charge. Positive charge and the negative charge are equal at a particular pH, and molecular weight used for comparing protein expression profile under different conditions. These two-dimensional usual gel also use the to know the protein profile at a particular conditions. Then next technique is Western blotting. This technique used to detect and quantify specific protein by separating them through gel electrophoresis and probing with antibodies specific to the target protein. Here in Western blotting, everyone know the test of HIV test. During HIV testing, first is ELISA, enzyme linked immunosorbent assay, and if the person having ELISA positive, then they go for the Western blotting analysis to confirm or quantify how much this gene related to the HIV is expressed by this Western blot techniques. And this technique is the only technique that quantify the protein expression. Then the protein microarray. These protein microarray are similar to DNA microarray, and these allow the study of thousands of proteins simultaneously to investigate protein interaction, function, and expression level. Because as we described, if a receptor ligand interaction, we can study that part in protein microarray. If we go for the expression analysis, then we can use also protein microarray. Then in X-ray crystallography and NMR spectroscopy, this X-ray crystallography and NMR spectroscopy determining the 3D structure of protein, that is secondary and quaternary structure of protein by this X-ray crystallography study, and this providing insight into their function and the interaction of this 3D structure of protein. Then we also label by different isotopes, a method to quantitative proteomics that uses isotopic labeling to compare protein abundance between two different samples. Because we label one patient of X protein and another patient of Y protein, then we go for isotope affinity tag, and then we characterize this expression by the different we know different uh, technique like MRI, CT scan. Here also we can study the particular protein is overexpressed in particular region or tissue or not. We also study by this proteome analysis techniques. Basically, we study the genome level and epigenome level, sometimes in transcriptomics, proteomics, and metabolomics. These are called the applied field of now research. These are called in applied field, and these are used for biomarker discovery as well as different types of disease condition like cancer cases or neurodegenerative cases, cardiovascular cases, liver failure. Every study nowadays, all the researchers use this omics approach for study of different types of diseases. Now, कार्य doubt अच्छी को doubt कर अच्छी को
सर जो प्रोसेस गुड़ा रहा सर जिनोमिक्स या प्रोटीमिक्स सर जो जानिया पे जो टेक्निक्स गुड़ा रहा सर सबु
ସେଗୁଡାକ ଆମେ
सबुर सहित
individually
गोटे
ଗୋଟେ ଗୋଟେ ହଁ ହଁ
आज्ञा
କୁହନ୍ତୁ।
आज्ञा ମୁଁ କହୁଥିଲି କି ଯେମିତି କି
ହୁଁ
ସବୁର ଗୋଟେ ଗୋଟେ ମାନେ ସ୍ପେଶାଲିଟି କିଛି ଏମିତି ବତେଇ ଦେଇଥାନ୍ତେ କି ଯେମିତି ଜଣା ପଡିଯାଇଥାନ୍ତେ କି ଏଇ ଜିନିଷଟା ହେଲେ ଏଇ ପ୍ରୋସେସଟା କରିବାକୁ ପଡିବ ଧର ଏମିତି ହେଲେ ଏଇ ପ୍ରୋସେସଟା କରିବାକୁ ପଡିବ ତ ସେଇ ଟାଇପର କହୁଥିଲି।
ଓକେ, ସେଇଟା ଆମେ ଯେତେବେଳେ ଆମେ ପର୍ଟିକୁଲାର ଗୋଟେ ଗୋଟେ ଟେକ୍ନିକ୍ସ ପଢିବା, ସେତିକିବେଳେ ଆମେ ଡିଫରେଣ୍ଟ ପ୍ରୋସେସେସ୍ ସବୁ ପଢିବା, କୋଉଠି ଆମେ କେତେବେଳେ ୟୁଜ କରିବା, ବୁଝିପାରିଲା। ସପୋଜ ଆମେ ଏବେ କ୍ୟାନ୍ସର କେସେସ୍ କୁ ନବା। କ୍ୟାନ୍ସର କେସେସ୍ ଯଦି ଗୋଟେ ଏକ୍ସାମ୍ପଲ ଆମେ ନବା, ଆମେ ଯେମିତି ଡେସ୍କ୍ରାଇବ କଲେ କି P53 ପ୍ରୋଟିନ ସମସ୍ତେ ଜାଣିଛନ୍ତି। P53 ନା ଜାଣିନାହାନ୍ତି? ଜାଣିଛନ୍ତି ନା ଜାଣିନାହାନ୍ତି? P53
ସାର୍ ଜାଣିଛି।
ରେଟିନୋବ୍ଲାଷ୍ଟୋମା ଜାଣିଛନ୍ତି?
ସାର୍
ଏ
ଆଉ ଟିକେ
ପ୍ରୋଟିନ ଦିଟା
କହିଦିଅନ୍ତୁ।
ଜାଣିଛନ୍ତି ନା ନାଇଁ? ରେଟିନୋବ୍ଲାଷ୍ଟୋମା P53। ସପୋଜ ଏଇ ଯୋଉ ଜାଣିନାହାନ୍ତି ଯଦି ଜାଣନ୍ତୁ, ଏଇ ଯୋଉ ରେଟିନୋବ୍ଲାଷ୍ଟୋମା and P53 proteins, these are called tumor suppressor genes। ଏମାନେ କଣ କରନ୍ତି ନା ସବୁବେଳେ ଆମ ବଡିରେ ଯଦି କିଛି ଏରର ହେଲା, ଇଏ ତାକୁ ରେକ୍ଟିଫାଇ କରିକି ଟ୍ୟୁମରକୁ ସପ୍ରେସ କରିକି ରଖନ୍ତି ବାय ସେଲ ସାଇକେଲ ରେଗୁଲେସନ। ସେଲ ଯେତେବେଳେ ଡିଭାଇଡ କରେ from one cell to another cell. During that process, these two proteins checks any error will happen in our genomes or in our DNA. Then it will allow to go to the next steps for division. If these proteins are mutated or changes or post-translational modified, then they will not check. They will not check in our cell cycle checkpoints. Then it will go for dividing, unlimited or on-program division. This unlimited or unprogrammed division will leads to and give rise to a tumor species. By this expression of this tumor p53 protein or retinoblastoma protein, we can know how the cancerous in different grade conditions by this expression. Like in breast cancer, we can study BRCA proteins, BRCA genes. We can study by this mass spectrometry study. If you study the single nucleotide polymorphism, suppose in forensic cases, we can use this next generation sequencing method. At this particular time, we can use this next generation sequencing method. If you go to the analysis of a particular one gene, if we analyze particular one gene, then we go for that polymerase chain reaction. These are the main and major techniques of genome analysis or proteome analysis. These techniques will provide any diseases cases like major diseases. Nowadays, lifestyle factors, we are targeting the lifestyle factor diseases means PCOS, polycystic ovarian syndrome, diabetes, blood pressure, because these are in a category that is lifestyle diseases, because due to the changes in our lifestyle, these diseases are common. So to study these type of diseases, we can use this mass spectrometry study, and this spectrometry study will give rise to what is the level of different signaling pathways or signaling molecules causes this hypertension or any diseases like liver failure, kidney failure, or cardiovascular disease. We can use for in pathway analysis. If we go to the mutation analysis, then we go for this genome analysis techniques. If we go to the enzymatic analysis, then we go for the proteome analysis technique. If we go to the signaling pathway analysis, then we go for only proteome analysis techniques. If we go for uh, deletion of a gene, duplication of a gene, any gene or DNA. If we're talking about any gene or DNA or chromosomal changes, then we can study by genome analysis techniques. If we particularly target signaling of a particular pathway or a cell or in any metabolic enzymes, then we will go for only proteome analysis techniques. These are the basic difference. Next ଆଉ କାହାର ଅଛି? ନେକ୍ସଟ ଆଉ କାହାର ଡାଉଟ ଅଛି?
ସାର୍ ଯୋଉ ସିକ୍ୱେନ୍ସ କମ୍ପ୍ଲେକ୍ସିଟି ସେଟା ଟିକେ ବୁଝେଇଦେବେ।
ସିକ୍ୱେନ୍ସ କମ୍ପ୍ଲେକ୍ସିଟି ନା ଜିନୋମ କମ୍ପ୍ଲେକ୍ସିଟି?
ସିକ୍ୱେନ୍ସ କମ୍ପ୍ଲେକ୍ସିଟିଟା।
ସିକ୍ୱେନ୍ସ କମ୍ପ୍ଲେକ୍ସିଟି ଆମର ଯୋଉଟା ଆମେ ପର୍ଟିକୁଲାରଲି କହୁଛେ, ସପୋଜ ଗୋଟେ ପର୍ଟିକୁଲାର ଅର୍ଗାନିଜିମରେ ଗୋଟେ ଲାର୍ଜ ସାଇଜ ର DNA ଅଛି, ଆଉ ଗୋଟେ ପାଖରେ ସ୍ମall size DNA ଅଛି, but that doesn't mean ଯାହା ପାଖରେ large size DNA ଅଛି ସିଏ ବେଶୀ ପ୍ରୋଟିନ ପ୍ରୋଡ୍ୟୁସ କରିବ, but ଯାହା ପାଖରେ small size DNA ଅଛି ସିଏ ମେ ବି ଟୋଟାଲ ସବୁ ଯେତେ DNA ସେଗମେଣ୍ଟ ଅଛି ସବୁ DNA ସେଗମେଣ୍ଟ ମେ ବି ପ୍ରୋଟିନ ପ୍ରୋଡ୍ୟୁସ କରିପାରେ, but large size DNA ସେଗମେଣ୍ଟ ତା ପାଖରେ ଥିଲେ ବି ସିଏ ସବୁ ପ୍ରୋଟିନ ପ୍ରୋଡ୍ୟୁସ କରିପାରିବନି। So these difference causes the genome or sequence complexity of DNA. For an example, ଯଦି ଆମେ ନବା ଇକୋଲାଇ କେसेस ନବା କି ହ୍ୟୁମାନ କେसेस ନବା, ଇକୋଲାଇ ରେ ଯେତିକି ଜିନ୍ସ ଅଛି ତାର ଜଙ୍କ୍ DNA, junk or intron region or transposable region are very less, but ଯଦି ଆମେ ହ୍ୟୁମାନ ବିଙ୍ଗ ପାଖକୁ ଆସିବା only 4% are translated to form proteins, but 96% are 96 to 98% are junk DNA or transposable region or intron region. We are more complex. ତ ଆମେ ସବୁ ପ୍ରୋଟିନ ପ୍ରୋଡ୍ୟୁସ କରିପାରିବାନି, ଆଉ ଆମ ପାଖରେ ବହୁତ ସାରା ରିପିଟେଟିଭ ସିକ୍ୱେନ୍ସ ଥିବ, ଆଉ ଆମ ପାଖରେ ବହୁତ ସାରା ଇଣ୍ଟ୍ରନ୍ସ ଥିବ ଯୋଉଟା କି ଆମର ଯେତେବେଳେ ପ୍ରୋଟିନ ସିନ୍ଥେସିସ୍ ହୁଏ ଆମେ ପ୍ରିଭିୟସ କ୍ଲାସ ରେ ଡେସ୍କ୍ରାଇବ କରିଥିଲେ, ଯୋଉଥିରେ କି ଇଣ୍ଟ୍ରନ୍ସ ଗୁଡାକ ଆମର ରିମୁଭ ହେଇକି ଏକ୍ସନ୍ସ ଗୁଡାକ ଯୋଡି ହେଇକି ଫଙ୍କସନାଲ ପ୍ରୋଟିନ ତିଆରି ହୁଏ। These are called complexity. ତ ଏଇଟା ଯଦି ଆପଣ ଜିନୋମ and the diversity of genome and different elements like this as we described the transposon elements like retrotransposon or DNA transposon, the signs, lines and ALU element, these are important. These we can use these for our study of the expression of our genes. For this we have to know the basics of the genome. Previously also we discussed about this the genomes of all prokaryotics and eukaryotic genomes, and these we can study by this genome analysis techniques like next generation sequencing. We can use different next generation sequencing for this particular genome analysis of different prokaryotes and eukaryotes, and the for proteins or proteomic analysis we can use this functional part of the proteins, that is that protein is affected, downregulated or not, or overexpressed or underexpressed, overexpressed or underexpressed, because all proteins are required for our body always in a basal level. If it crosses the basal level, that is suppose one is for an example basal level is 5, if it crosses the basal level of 5, then we called it overexpressed protein. If we uh, if we found less than the less than 5, then we'll call it this is underexpressed, and then we can describe underexpressed or overexpressed by this proteomic analysis techniques. ठीक ଅଛି, ତ ଏଇଟା ଆମର basic of diversity of genome and C value paradox and different techniques. In our next class we will discuss about individual techniques and how these techniques will be used in different disease cases or different genetic mapping or different genetic expressions. ଆମେ ନେକ୍ସଟ କ୍ଲାସରେ ସବୁ ଆକୁ ଡେସ୍କ୍ରାଇବ କରିବା different techniques को with suitable examples. ଠିକ ଅଛି, ଆଜି ପାଇଁ ଟିକେ ମୁଁ ଶୀଘ୍ର କ୍ଲାସ କ୍ଲୋଜ କରୁଛି କାରଣ ମୋ ଦେହଟା ଭଲ ଲାଗୁନି ଆଜି, ମୁଁ ସେଥିପାଇଁ କି ଆଜି ଟିକେ ଶୀଘ୍ର କ୍ଲାସ କ୍ଲୋଜ କରୁଛି।
Yes, sir.
ହଁ, ମୁଁ ସେୟାର କରିଦେବି
Thank you, sir.
PPT ଟା ଠିକ ଅଛି।
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Yes, sir. Thank you, sir.
ହଁ, thank you।
Thank you, sir.